Okay, welcome dear learners. In this video, we will learn about some important questions and their explanations. Some students have some difficulties to understand these questions and their solutions. So we will try to uh, give clarity on them. Among that, the first question is, glycogen is a branched chain polymer of alpha D glucose units in which chain is formed by C1 to C4 glycosidic linkage, whereas branching occurs by the formation of C1 to C6 glycosidic linkage. Structure of glycogen is similar to. So that is the question. Actually, glycogen is a polymer. As you know, the meaning of polymer is a monomers will form the polymer. Here the glycogen is a polymer, and that glycogen is made up of D glucose, alpha D glucose. Those are the monomers here. So in that question itself they gave the answer, some of the answer, that is each monomer of the glycogen is held by alpha 1,4 glycosidic linkages, and the branching site where the branch attaches to the native chain of the glycogen molecule are held together by alpha 1,6 glycosidic linkages. So the similar structure is present in the four options. We have to identify that one. Here the first one is amylose. Amylose means starch. Actually, starch is also a polymer, and it is made up of glucose monomers. Starch is also called as amylose. And the starch is a water soluble molecule. Here glycogen is called as animal starch because the excess glucose that is present in human body is stored as a glycogen in the liver and muscles of the animals. Hence it is called as animal starch. Whereas starch is the main storage molecule in the plants. That is, plants synthesize the glucose by the process of photosynthesis, and those glucose monomers are held together by a alpha 1,4 glycosidic linkages, and they will produce the starch or amylose molecule. So here amylose is not a correct option as it is not a branched polymer. So then cellulose is also present in plants. Cellulose also a polymer of a glucose molecule. The main difference between the starch, that is amylose, and cellulose is amylose or starch is made up of alpha D glucose molecules, whereas cellulose is made up of beta D glucose molecules. There is a conformational change present between those two molecules. So we have to identify the point. Some questions may also be acts as which one is odd one. They will give the options amylose or starch, cellulose, like that. So amylose or starch is also a polymer, and cellulose also a polymer, but amylose or starch is a polymer of alpha D glucose, whereas cellulose is a polymer of beta D glucose, and those two are not correct options for this question. Finally, fourth one is a glucose. Glucose is a monomer, not even at all a polymer here. So the correct option is amylopectin. Amylopectin is a polymer of alpha D glucose, and this is insoluble in water as in glycogen. In amylopectin also, those two glucose molecules, alpha D glucose molecules, are held together by alpha 1,4, that is first carbon and fourth carbon of the glucose molecules are involved in the glycosidic bond formation, and the branching site alpha 1,6 glycosidic linkages are present. That is the correct option for the first question, that is amylopectin is the correct answer. And then another difficult question is fifth one. Proteins are found to have two different types of secondary structures, alpha helices and beta plated sheet structure. Alpha helical structure of a protein is stabilized by. Generally, proteins are also a polymer molecules, Here the monomers are called as amino acids. So different amino acids, about 20 amino acids build all the proteins present in our body. The proteins mainly have 4 levels of structural organization. They are called as primary structure, secondary structure, tertiary structure and quaternary structures. Primary structure means the amino acid sequence, the protein by which amino acids are producing that protein. The primary sequence, the primary linear sequence of those amino acids are called as primary structure, and these amino acids held together by specific bonds, they give up to some different structures to the protein, and those second level organization of the protein is called as secondary structure. And mainly proteins contain 2 types of secondary structures. First one is alpha helix and second one is beta plated sheets. Alpha helixes are present in, for example, in keratin. Keratin contains alpha helical structure, and those alpha helical structure in protein is stabilized mainly by the hydrogen bonds between the amino groups of hydrogen and carboxyl groups of oxygen atoms, carboxyl groups of hydrogen atoms. So that is the 5th question answer. And then the 7th question is, which of the following acids is a vitamin? Here first option is aspartic acid. Aspartic acid is an amino acid that not going to be acts as a vitamin. And second one is ascorbic acid. Actually ascorbic acid is also called as vitamin C, because vitamin C is most abundantly present in citrus fruits like lemon, orange, like that. Ascorbic acid is coenzyme or cofactor for many enzymes in our body, and it is mainly involved in the cartilage formation, tissue repair, and some of the important metabolic reactions. So the correct option is ascorbic acid for 7th question. And then here 15th question, 3 cyclic structures of monosaccharides are given below. Which of these are anomers? Which of these are anomers? Actually anomer means the 2 molecules differ in particularly at a specific carbon atom. So anomers means they contain similar structure, but they only differ in at a specific carbon atom, particularly C1 and C2. C1 and C2. At those positions, they will have some different functional groups there. They are called as, that's why they are called as anomers. Anomers may be 2 types. These 3 options are given are hexoses because they contain 6 carbon atoms. Hexoses may be again 2 types. One is aldoses and second one is ketoses. Aldoses means at C1 position, at carbon 1 position, aldoses contain aldehyde group, that is CHO. Ketoses means at C2 position, they contain carbonyl group, that is CO, keto group. So these are they differ particularly at 2 carbon atom positions at C1 and C2. If we are talking about the hexose, that is glucose, at maybe in C1 position. If we are talking about the another hexose, that is fructose or a ketose, that will be differ in C2 position. So the correct answer is, we can see here our 15th question. One second. So first option is correct. 1 and 2. So you can see here at second carbon right side hydroxyl group is present OH, but see in second option at C2 carbon right side is a hydrogen atom and the hydroxyl functional group is present at left side. They differ in C2 position here. Observe this. Here hydroxyl group is present at right side and here hydroxyl group is present at left side. Like that, the molecules which differ in particularly C1 and C2 positions are called as anomers. And then next question, I think 22. Which of the following carbohydrates are branched polymer of glucose? If you observe this, all of these are polymers of glucose. As we explained at first question, amylose or starch is also a polymer of glucose. Amylopectin is also a polymer of glucose. Cellulose also a polymer of glucose, and glycogen also a polymer of a glucose. But your question is, which of the following carbohydrates are branched polymer of glucose? Which molecules have here branches? Actually, glycogen is a beta D glucose polymer of glucose. Amylopectin is a alpha D polymer of glucose. So here that both two options are also correct, that is second one and fourth one. We will see what is the answer they given. For 22nd question, 10, 19, 22. Both 2 and 4 are correct. Both 2 and 4 are correct. That is correct. Another question they will ask like which of the following polymer is made of alpha D glucose? Then we have to answer as starch or amylose. Which of the following polymer is made of beta D glucose? Then we have to answer as glycogen is a polymer of beta D glucose. And then again another important or another tricky question is 9th one. Nucleic acids are the polymers of. Actually, nucleic acids are polymers of nucleotides. Monomers are called as nucleotides. That is the correct option is second one, nucleotides. Then we have to know what is the meaning of nucleotides. Here nucleotide means each nucleotide is again made up of 3 components. That is, first one is a ribose sugar, and second one is a phosphate molecule, and third one is a nitrogenous base. So with all these 3 things a nucleotide may be formed. Those are, I will tell again, ribose sugar, phosphate molecule, and a nitrogen containing base. These 3 molecules combine together and form a particular nucleotide. And nucleic acids like deoxyribonucleic acid DNA or ribonucleic acid RNA, both are made up of these nucleotides. The fundamental difference between these 2 nucleotides are in DNA, that is deoxyribonucleic acid, the ribose sugar may be a deoxyribose. In the sugar will be ribose itself. So this is the fundamental difference. Then come to the nucleotides. Nucleotides are made up of ribose sugar, either it may be a deoxyribose in DNA or ribose sugar in a RNA. So ribose sugar is held together in one side by phosphate molecule and in another side with a nitrogenous base. So again, these nitrogenous bases are differently, mainly two types, that is one pyrimidines and purines. Cytosine and thymine are called as pyrimidines. Adenine and guanine are called as purines. So these all things are peptide means it contains three moieties. First one is ribose sugar, second one is phosphate molecule, and third one is nitrogenous base. In nucleic acids, either it may be in DNA or RNA, ribose sugar and phosphate molecule will form the sugar backbone in the nucleic acid structure. In between that sugar phosphate backbone, nitrogenous bases are held by hydrogen bonds. Here nitrogen bases are types. One is pyrimidines and second one is purines. Adenine and guanine are called as purines. Cytosine and thymine are called as pyrimidines. So that is the answer for second one. So second option is the correct answer for 9th question. And then finally, I think which one of the following a purine base? As I explained now, which one of the following are purine bases? Purine means there are two purines are present. One is adenine and second one is guanine. So here the option is, they gave both the options. They gave adenine and guanine. So the correct option is 1 and 2. We will observe answer for 27. So 27, 1 and 2, that is correct. Here the adenine and guanine molecules are called as purines, whereas cytosine and thymine molecules are called as pyrimidines. If you observe the question again, we will come to know another important point, that is they gave the fourth one as uracil. Uracil also a pyrimidine. Cytosine also a pyrimidine. But in DNA, thymine and cytosine pyrimidines are present, but in RNA, that is ribonucleic acid, instead of cytosine, the cytosine is replaced by the uracil. So RNA contains the pyrimidine uracil, the DNA contains the pyrimidine cytosine. This one also a fundamental difference between deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. So DNA contains cytosine, whereas RNA contains uracil, and both these cytosine and uracil are called as pyrimidines. So we conclude that actually nitrogen bases are 5 different types. First one is adenine and guanine. Those two are called as purines, and remaining 3 are called as pyrimidines. The pyrimidines are thymine, cytosine, and uracil. Thymine and cytosine are present in DNA, but instead of cytosine, uracil is present in RNA. These are the important questions. Hope you will understand. Because of some technical issues, I am unable to show all the PPTs. Hope you will understand. Thank you very much.